السلام عليكم طلاب. اليوم راح نبدي المحاضرة الثالثة اللي تتعلق بالسبيشال سينس smell and taste. For objective learning to this lecture, describe the anatomy of the nasal cavity and its sensory function in smell. And second, we are talking about the sense اللي هو يتعلق بالتيست. So describe the anatomy of the tongue and its sensory function in taste.
Now, smell and taste are working together because it's represented about the chemical sense that will be responsible for giving your smell and taste. So these two tastes are closely related to each other. Both senses are strongly and tied to primitive emotional and behavioral function of our nervous system. Because the smell and taste are closely related together, it is dependent on chemical substances that will be sent by the receptors and sent into the nervous system. Taste and smell cell are closely together, and the only cell in the nervous system that are replaced when they become old or damaged. Another cell cannot be replaced, as you know, because the nervous system cannot be replaced. Another cells, when it will be dead, the cell cannot be replaced by another. But in this senses, taste and smell, this is the only cell can be replaced when it damaged or be old. So the smell or olfaction and taste gustation, complementary sense that lists now whether two substances should be served or avoided. Because it is represented or considered as by the receptor of the chemical receptor, it will be substances will be savored or avoided. The sense that will be dependent on the taste or smell, it will be should a sense be savored or avoided, dependent on the it is dangerous this taste, we avoided about it, or it is benefit, we must be go to take this. Now, what is mean of chemoreceptor? Or this taste are dependent on the chemoreceptor. This is used by the system, and this is chemical must be dissolved in aqueous solution to be picked up by the chemoreceptor. So the smell receptor are excited by chemicals, and these chemicals are dissolved in the nasal fluid, while the taste receptor respond to chemical dissolved in saliva. So this both smell this both taste اللي هي smell and taste have a receptor. In the smell are we excited by a chemical. This chemical substances by the ordinate are be dissolved in the nasal fluid and then send signal to the brain, while the taste receptor found on the tongue are represented by a chemical. This chemical substances when you are taste it are dissolved in saliva, and then the receptor of the cells inside the tongue will be send a signal to the brain about the taste. Now, the sense of the smell found in the structure of olfactory receptor means olfactory epithelium. So this is olfactory epithelium are organ of smell that will be located in the roof. Now it is the position of it. The position of olfactory epithelium, it will be located in a roof of nasal cavity, and this is covered superior nasal conchae. This is occur in the roof of the nasal cavity and covers superior of nasal conchae. Now this is olfactory epithelium, have olfactory sensory neurons, olfactory sensory neurons and bipolar neurons with radiating olfactory cilia. This is will be supporting cell surround and making a cushion, making this is supporting cell surround and making cushion olfactory receptor cell. You can see in this picture the rotate of inhaled area, how can be, this is smell, this is odorant, can be enter to the nasal conchae, and when it enter to the nasal conchae, it will be reach to the olfactory epithelium, and olfactory epithelium have the up of it have a olfactory bulb. This is olfactory bulb send or have olfactory neuron cells that will be communicate with the olfactory tract, and then this is olfactory tract reach to the special senses will be attached to the nerve cell in the brain stem and send signal to the brain. Now in this picture you can see the relationship between taste and smell senses. The eat or the food, you can smell it before your taste. When you enter in your mouth, the food can be tasted by the special nerve cells found on your tongues, and this is special nerve cell after tasted the food send a signal to the brain stem. And also in the same time when you taste or before you eat, the aroma of the food can be entered on your conchae of your nose, and when it reach to the epithelial roof, it will be have it will be found the olfactory bulb. Olfactory bulb have signals or have a receptors neuron cells. This is will be send a signal to the brain to communication with the same time at the taste where you ate your food, and this is send a signal to the brain to be explain this signal from your taste or odor net. Now activation of the olfactory bulb. Olfactory bulb or olfactory mucosa is divided into 4 zones. You can see we have 4 zones: zone 1, zone 2, and zone 3, 4. Each zone of this are a variety different of receptor. This is have a variety different of receptor, and a specific type of receptor are found in only one zone. You can see different color in these types of zones. Each zone have a different color from another zones, and have olfactory sensory neurons. This is olfactory sensory neurons found only in only one zones with different color. So odorant tend to activate neuron within a particular zone. So you can see the olfactory bulb and the different zone of olfactory bulbs. Each one of different zone have a sensory neurons, and this is specific sensory neurons have found in only one zone, so it will be activate neuron with a particle zone. The olfactory receptor are located in the specialized portion of the nasal mucosa. This is receptor found in a specially portion of nasal mucosa we are called about the olfactory mucous membrane, and this is located on the roof of on the nose near to the septum. You can see in this picture the different steps when you are odorant are enter in your conchae by the and reach to the olfactory nerve passing during the olfactory bulb. So we have first odorant bind to the receptor. This is small molecules are bind to the receptor found in your in your nose, and this is will be olfactory receptor. When it odorant are enter in your nose, olfactory receptor cell are activated, and this is will be send electrical signal. So this is electrical signals are relayed by converged axon, and this is electrical signal are conveyed by the relayed axons or converged axon. When it enter from the olfactory receptor cell reach to the olfactory bulb. As we said, the olfactory bulb have receptors, and this is odorant are entered by the receptor of the odorant in your nose, reach to the olfactory bulb, and then send signal to the convergent axon. After will be sent, it will be transmit by the signal to the region of the brain, and region of the brain will be analyzes this smell. It will be harmful or it will be useful to be inside in your when you enter inside in your nose, and then pass to the olfactory bulb and sent by the nerves inside your brain. Now the physiology of the smell. How can smell be transmitted, or how can be it will be entered by the opening channel, different channel in your body. So this is channel I'll be responsible for smell. Olfactory receptor responds by several different odorant causing chemical. When bound to ligand this protein, when bound to the ligand this protein, initiate a G protein mechanism. This is G protein mechanism or will be uses a cyclic IMP. This is as considered a second messenger. Now cyclic IMP are by the activation of the action potential are open the sodium channel and calcium channel. This is causing depolarization of the receptor and the trigger as an action potential in the presence of adenosine triphosphate. The mechanism of stimulation of olfactory receptor. Odorant are binding to the specific membrane receptor protein. Now, this specific membrane protein will be activation or opening of G protein stimulation to G protein. The stimulation of G protein will be activation the cyclic AMP and binding of this cyclic AMP to special canal. This special canal will be opening a special of influx of sodium and calcium. This will be influx of sodium and calcium, making a depolarization of the trigger channel to be opening of this neuron cells by the action potential, and then will be current flow to membrane of depolarization by the calcium or other channel or will be opening during this stimulation of olfactory receptor. And finally, clinically, we have a homeostatic imbalance in your odorant on your olfactory sense. It will be known as anosmia. Now, this is olfactory disorder are common or most result from maybe from head injury. Now, head injury means that tear olfactory nerves. Also, maybe by after effect of nasal cavity inflammation. So, an inflammation in nasal cavity may be lose of the your smell or your sense of smell. Maybe by neurological disorder such as Parkinson's disease. Also, it occurs anosmia by the olfactory hallucination. Now, olfactory hallucination are related caused by temporal lobe epilepsy, special temporal lobe of epilepsy, it will be involved. This is the special lobes epilepsy, the temporal of it will be involved in the olfactory cortex. This is the final slide of this lecture. I hope you understood this lecture, and if you have any question, you can ask me at any time. Thank you for listening in this lecture. Now, taste sensation. This is include the taste bud, and most of the 10,000 or so taste bud are found on the tongue. So taste bud are found in papillae of the tongue mucosa, while papillae come in three types: filiform, fungiform, and circumvallate. Fungiform and circumvallate papillae contain taste bud. Now, this is sense of taste are mediated by taste receptor cell. This is a bundle in a cluster called taste bud. So, taste receptor cell is a sample of the large number of small molecules. This is sensation of taste lead to the center or reach to the center in the brain stem. In humans, we can see these types of taste bud, and this is taste bud are most common of epithelial of tongue. Now it's called as papillae. This taste bud themselves are too small to be seen without a microscope, but you can see the papillae. It can be observed by the close of in the tongue surface. But now each ground-shaped taste bud consists of three major layers, and this is major cell types. Consists of the three major cell types. Now, mean supporting cell that will be in solute the receptor, and basal cells responsible for dynamic stem cells, while gustatory cells responsible for taste. Now, about the basic sensation taste, we have 5 basic taste sensation, which means sweet, sour, salty, bitter, and umami. Now, sweet, which means sugar, saccharin, alcohol, some amino acids, and sour, which means hydrogen ion in solution, while salty, metal ion, and bitter-alkaloid such quinine and nicotine, caffeine, and umami. This is basic taste sensation include in amino acid, glutamate, and aspartate. Example like a meat. So this is sense of taste have a receptor for a large number. This is sense of taste have a receptor of large number of a specific chemical substances, a specific chemical. It will be identified to contribute to the reception of the taste, and this will be sweet, umami, salty, sour, bitter. Each one have a taste, special tastes that will be recognized. Any taste, if you have a bitter, umami, salty, sour, or another taste in occur in the tongue. Now, physiology of the taste. How can be understood the physiology of the taste? By dissolved to be able the taste to chemical we must be dissolved in saliva, physiology of the taste, to be able to taste a chemical. It must be dissolved in the saliva and diffuse into taste pore, while finally contact gustatory hairs. This is the basic physiological taste that will be occur when the taste are recognized. A chemical substances must be dissolved in saliva, must be diffused into taste pore, and finally contact to the gustatory hair. Now, activation of taste receptor. Beginning, binding of food chemical. This will be depolarized cell membrane of gustatory epithelial cell membrane. And after this will be causing release of neurotransmitter. Now, when neurotransmitter is released, that will be bind to dendrite of sensory neuron and initiate a generator potential that will be to action potential. After this, different gustatory cells have different threshold for activation. Now, beginning, binding this food for the chemical lead to the depolarization of the membrane of gustatory epithelial cell, and according to this, that will be release of the neurotransmitter. After release of neurotransmitter, this will be bind to the dendrite of sensory neuron and initiate a generator potential. This is lead to the action potential. While action potential are occur, different gustatory cell have different threshold for activation. Now, taste as a small to medium size molecule dissolve in saliva. When you are taste anything, smaller, medium or size molecule on the tongue, that will be dissolved in saliva, and after it dissolve in saliva, reach to the or dissolve in the or enter to the taste pore. Bind taste pore have a receptor, so this will be bind to the receptor in cilia, and this is receptor have a potential lead to the action potential. Now, taste transduction. Gustatory epithelial depolarized by salty taste is due to the sodium influx that directly cause depolarization. Now, if you have a taste of salty taste, this will be causes depolarization because of the sodium reflex. While in sour taste, this is due to the hydrogen acting intercellularly by opening channel to allow other action of the enter, allow other action to enter. Unique receptor for sweet, bitter, and umami, but all are coupled to G protein. So any activation causes release of stored calcium that open action channel. When this channel are open, this is causing a depolarization and release of neuron transmitter ATP. You can see this explanation in the next slide as a diagram or as a picture. Chemoreceptor or gustation and olfaction. Chemoreceptor, which mean chemical substances. This chemical substances are dissolved in a mucus. When it dissolved in a mucus, gustatory epithelial cell depolarized. This depolarized means G protein as linked receptor, and all smell and some taste. G protein are linked receptor and all smell want some of taste. What is the taste of G protein linked receptor? Sweet, bitter, umami. This is a sweet, bitter, umami are release of G protein linked receptor and channel linked receptor and of other cell channel linked receptor of other some taste like sour or salty. So as we said, salty taste due to the sodium influx, while sour taste are due to the hydrogen acting. You can see in this picture how can G protein linked receptor in sweet, bitter and umami, and how can the hydrogen or how can the channel linked receptor and other taste like in sour and in salt. We can see in salt sodium influx that directly causes depolarization and salty taste, and in sour taste you can see the hydrogen acting that will be opening channel to allow other quotient. How can be opening channel to allow other quotient to be enter inside this test. Now, gustatory pathway. Three main cranial nerve pair carry taste impulse from the tongue to brain. Gustatory pathway. Three main cranial nerve pairs carry taste impulse from tongue to brain, beginning by the facial nerve. Facial nerve carries impulse from the anterior 2/3 to tongue, and glossopharyngeal carry impulse from posterior 1/3 and pharynx. Vagus nerve transmit from epiglots and lower pharynx. And lower pharynx. Fiber synapse in the solitary nucleus of the medulla, then travel to thalamus, and then to gustatory cortex in the insula. Hypothalamus and limbic system are involved, allowed to determine appreciation of the taste. This is can see in the figures or in this picture how the physiology of the taste or how the mechanism of the taste are occur to be reached in the brain. You can see the transduction process by stimulus energy and converted by nerve impulse. So each taste appear to have a mechanism. Each taste appear to have a mechanism like salt. As we said, it will be released the sodium influx through sodium channel by allow to another calcium influx, and sour is mediated by hydrogen, appears to action taste cell in 1-3 way, appears to action of another taste by a 3-way, as we said, directory entering or opening channel or other quotient by enter other substances, and bitter, sweet and umami. This is 3 by opening G protein channel, by opening G protein channel mechanism that will be send a message by depolarization, increasing intracellular by increasing the intracellular substances maybe bitter, maybe sweet, maybe sour. It will be maybe bitter, maybe salty, maybe sorry, not bitter, sweet, or umami. That will be send a message to depolarization and opening channel. Clinically, we have a homeostatic imbalance. So what or when taste or disorder occur? This is occur less common than disorder of smell because taste receptors are served by three different nerves. Taste receptor, it is less common than of the smell because it is served by three different nerves. Not likely that all nerves would be damaged at the same time. So causes of taste disorder include upper respiratory tract infection, or maybe head injuries, maybe chemical or medication, or head and neck radiation for cancer treatment. This is talking about the taste sensation. I hope you understood, and if you have any question, you can ask me at any time. Thank you for listening and enjoy to it.
بالنسبة للسؤال اللي يتعلق بمحاضرة اليوم راح يكون what is the mechanism of smell and what is the mechanism of taste. As a recap for this lecture, smell and taste are closely related to each other. Also, smell and taste complementary senses that let us know whether a substance should be saved or avoided. Olfactory mucosa is divided into 4 zones. Olfactory receptors respond to several different odor-causing chemicals, and taste buds are found in papillae of the tongue mucosa. There are 5 basic taste sensation هاي اللي هي الساور واليونيكيول الساور والبتر والسويتس. هذين الخمس sensation اللي هنا يمكن أن نتذوق بهن. والgustatory epithelial cell depolarization caused by salty taste, sour taste, and receptor for the sweet, bitter, and umami. Thank you.